Hi, hello, everyone, and welcome to our Science Cafe tonight, organized by the UMass Chan Postdoc Association and UMass Chan Medical School here in Worcester, Massachusetts. Our event tonight is on the topic of the neuron degenerative disease, amyotrophic lateral sclerosis, or ALS. Before we introduce our speakers, I want to quickly remind everyone that the views in the speakers' presentation are solely on the presenters and not necessarily those of Massachusetts Chan Medical School. Also, our science cafes are recorded and will be uploaded to our YouTube channel here if you want to share the seminar with your friends and colleagues. And also, please follow our social media channels listed in the chat box if you would like to stay up to date with our events. Also, at the end of the talk, if you have any questions, please type it in the chat box or click the raise hand button, and we can queue you after the talk. We will also ask you your question at the end of each presentation. So our first speaker today is Aparna Sreeram. Aparna is currently a graduate student in John Lander's lab at UMass Chan Medical School. Aparna has a bachelor's degree in biotechnology from India and firstly came to Massachusetts to pursue her final undergraduate year in the semester abroad program at Harvard Medical School and Boston Women's Hospital. Since then, she stayed in Massachusetts and earned her master's degree at the Boston University. She was firstly introduced to ALS at BU, and her thesis focused on the role of UBQL2, which is a gene mutated in ALS patients. And she is continuing her ALS research work here at UMass. She is passionate about translational neuroscience research, and particularly in identifying new therapeutic targets of them. Outside the lab, she practices yoga and meditation, which she has trained in institutes across India and the United States. She also likes reading legal crime thriller novels whenever she gets some time off the lab. So, Aparna.
Hey, thank you for that wonderful introduction. I'll go right ahead and share my screen here. Um, Hope everything looks fine. Okay. Um, Hey everyone. So today I'm going to be talking to you about the most complex organ of the human body, the human brain, and I'll introduce you to a disease that affects the brain cells in the brain called ALS, and also talk about what happens to the body during ALS. So to really understand the complexity of the brain, we need to first understand the basic fundamental building blocks that makes it up, which are the human cells. So humans are made up of trillions of cells, each having their own structure and function depending on where they are localized in the body. So the cells in the brain are called as nerve cells or neurons. So these neurons are really unique because of their structure. They have these long projections called axons that help them to connect with their neighboring cell and help in transmitting information throughout the brain as well as throughout the human body. So these neurons are present all over the brain. And here is a very simplified diagram of the human brain. And these are divided into these lobes, as you can see, color coded here. And each part of the brain is responsible for controlling each body function. But today, I'd like to talk more about the motor control region of the brain. And as the name suggests, it's involved with all the motor movements. So the motor control region is also called as the motor cortex. And this motor cortex functions by helping in planning and executing any voluntary movements. So what this basically means is that every time you're thinking about moving your hands or walking down the street or talking or eating and even breathing, these motor cortex are actively planning.
Sorry, Aparna, we can't hear you.
Is it okay now?
Okay.
Okay, sorry about that. Yeah, so because this motor cortex has these neurons that originate from up here called as the motor neurons, and they travel down the brain and into the spinal cord and reach these muscles. And this transmits signals that helps in muscle movement. So when we actually zoom into this region, what you can see here is that these motor neurons send out these long axons that actually get into the muscle and help in muscle movement by transmitting signals. But this is a really simplified diagram. It's more complicated than this in that this is actually a network of neurons operating in this area, and where each of the neuron talks with their neighboring cell, and as well as multiple neurons get into this muscle at different points, helping in muscle movement and coordination. Uh, so, why am I talking about these motor neurons? It's because in amyotrophic lateral sclerosis, also called as ALS, these motor neurons die, as a result of which the muscle is unable to function, and this results in diminished muscle contractions. So, ALS is a rapidly progressive disease. And although it's considered to be rare, it is more prevalent in that every 90 minutes, someone is diagnosed with this disease. And the average age of onset is between 40 to 70 years. But there are also patients who get ALS in their late 20s and early 30s. ALS does have some family history associated with it, but most of the cases occur without a family history. And the average age of survival is between 2 to 5 years after diagnosis. And it's predicted that there's going to be an overall increase of 70% in ALS in the next 15 years or so. But the most sad part of this disease is that there is still no cure for ALS. So, based on uh, what I just told about the motor neurons and ALS, which of the following do you think could be an early symptom of ALS in these patients? Um and if you could run that poll. Yeah, we're still at 70%. Okay. Yeah, so yeah, and these people have symptoms that affect their muscles, so they have muscle weakness. Uh, and because these motor neurons progressively die, they have difficulty walking and speech problems, and ultimately breathing, which becomes the cause of death in these patients. So, what are we doing towards a cure for ALS? So, if you remember a decade back, ALS became really famous for the Ice Bucket Challenge, and that raised millions of dollars towards this disease. And very fortunately, a lot of this amount went towards research, and more fortunately, a lot of this money went into to labs at UMass, and you can see the professors at UMass taking the Ice Bucket Challenge here and also finding new things about ALS. And a decade later, what we can say is that this has dramatically improved our understanding of ALS as well as accelerated drug discovery. Um, and this is and the overall money raised from the Ice Bucket Challenge helped us ask more basic and critical questions like why do motor neurons die in ALS? And as a first step, we wanted to look into these patient genes. And before I move further ahead, I want to take a step back and talk a little bit about what are genes. So inside every cell, there is the DNA genetic material that's passed on from your parents called DNA. And this is tightly packed into these chromosomes. And genes are segments of DNA containing information about that particular trait or a characteristic. But this information is uh, not easy to understand because it's coded. it has these nucleotides called adenine, thymine, guanine, and cytosine, also called as AT, G, and C. And for any gene to be expressed, it needs to be converted from this DNA state to an intermediate mRNA and in a process called transcription. And this mRNA needs to be converted to proteins in a process called translation, which are and proteins are the real players for expressing a gene. And Karin has a wonderful analogy explaining this, so I'll just let her do that. But getting back to ALS, um, how can reading this gene expression really help understand ALS? So what we can do now is that we can conduct this large study where we group all the patients together and healthy individuals and just read this code A, T, G, and C in a process called genotyping and compare the patients and the healthy individuals. And once we have this, we can then use statistics to calculate any changes that we see at a higher frequency in these patients compared to healthy people. And by doing this, what scientists have found is that there are different types of gene changes or gene mutations in these patients. One being these insertions, where a nucleotide is being inserted into the gene. Here, A is being inserted. And the other type of mutation is substitution, where a particular nucleotide is substituted for another one, a G becoming a T here. And lastly, there is something like deletions, where these nucleotides are deleted from the gene. And all of this leads to defects in patients that we see. So over the years, there have been several gene mutations identified contributing to ALS, as you can see in this graph here with the years in the y-axis and the genes on the x-axis. Um, and the size of the circle here represents the number of people living with that mutation. What is really striking here is this big, bright yellow circle uh, showing that there are more people living with C9orf72 gene mutations, and that's more commonly found in these patients. So what does this mutation do? So in a normal cell, there is this C9orf72 gene that makes normal proteins that and gets expressed. But in a patient with this mutation, it's an insertion mutation where now there is this hundreds of 6 nucleotides, 4 Gs and 2 Cs being repeated in these patients. As a result of which, it leads to these toxic RNAs and toxic proteins being made, and also leads to lower levels of the gene itself. But again, we haven't really addressed the question, how does this mutation affect the neurons? And to study that in detail, we first need to model the disease. And for doing that, we have developed stem cell models. So this stem cell model basically works by taking skin cells from ALS patients and converting them into stem cells. And by converting them, we reset their genes in that these stem cells can be converted into any type of cell, including the brain cells or neurons. And once we have these neurons, these neurons have the same mutation as what we see in these patients. And once we have them, we can now study them on a dish uh, and um, and look at why these motor neurons die, and further use drugs to test them and prevent them from dying, which can one day become into ALS specific drugs that can be used to treat patients. So this is a timeline from this stem cell phase to mature neuron phase over a span of around a month. And once we get these patient cells, what we can do now is look at their gene expression and ask what are the common genes that's altered in these patients. So we did that to our study, where we took 4 different ALS patients and looked at and took the stem cells and neurons and just read their gene expression in all these ALS patients. And what we found was that there were common genes altered, affecting neuron communication, transport in these neurons, as well as very early on, these cells begin expressing these cell death genes. So now that we have all this information, what can we actually do with it? So we then again go back to this diagram and look at drugs that can specifically target these genes. And down the lane, these can hopefully become into drugs that can actually be used to treat patients. And Karin has another great strategy for treating patients using another technology, which we'll talk about. But we hope that one day this could be an answer to ending ALS. And with that, I'd like to thank my lab and all the collaborators and my thesis committee for helping me with this research work. Thank you.
Well, thank you, Aparna. It was very good talk and very informative. So if you have any questions to ask Aparna, you can type in chat box or I can unmute you so that you can ask by yourself.
I have one question.
Mhm.
Can I ask? Hi, Aparna. Nice
Yeah.
talk. I just want to know that if diet and exercise, does it have any effect or environmental effect on the the fate of the neuronal cells? Does it change?
Yeah, that's an interesting question. If you remember, there's like 90% of the disease that we do not know the cause for. And most of it is actually uh, coming from environmental causes like diet and like exercise and all of that, because these are changes in like if you're like having extreme diets, or if you're not, if there is any environmental uh, exposure that you have, that has also been shown to lead to ALS. And there have been like a lot of research with respect to this. Like for example, there was one study that looked at pesticides leading to ALS, like symptoms in cells, and also smoking and other like intense exercise, or as well as like reduced exercise have been like shown to have some ALS like symptoms in cells. So yes, that's certainly something like people are researching about.
And follow-up question. Thank you for answering. I just want to know that if there is any genotype suggestion where you've seen post-transfer modifications in case of protein things, like any like suggesting any higher post-transfer modification happening in the protein level to see the effect on ALS, because that correlate with the if there is any mass spec or some type of suggesting about the protein level, because you mentioned about the insert, like you see the 100, like there's especially some genes got mutated having like insert of different sequence, but is it like I'm just curious.
Mhm.
That's what
Yeah,
my
so
question.
yeah, there are like uh I'm not sure about like post-translational modifications. I mean there are studies, but I don't think anything came up like very consistently between like all ALS mutants. But there are like these especially in C9, there are these proteins that's being formed that's usually like detected in patient samples. And these proteins are not like normal proteins. They are actually just toxic proteins that accelerate the disease. So they are actually being used as like biomarkers and stuff. But there are general uh, like neurofilaments that's being used in as biomarkers in the CSF uh, of these patients. But
Uh, Toxic mutant, you're talking about apoptotic marker induced, or what? Like for
no, they are like just stemming from the gene mutation itself. So because of abnormal RNA being produced due to the insertions, they have and those RNA are translated into like proteins. Uh, and these proteins are like dipeptide repeats.
so, just clarify myself. So the mutation happening in all the regulatory proteins or the essential proteins, and like all the mutation in the genotype, which type of more get high uh, uh, impact on the mutations? It's like, is it like there is range of mutations, but I'm just curious that is it affecting the regulatory or type of essential proteins which is necessary for cell like neurons to survive?
It's more like much more complicated than I think. It's because like these proteins they just build up in the cell. So we do not know if they are
Because
actually the
because what I'm curious about is that, as you know, that in cellular context there is also a machinery which makes sure that there is no mutation, like the mutation rate is goes down. But I'm just curious that if like maximum, because you mentioned there is motor protein, there is cytoskeleton, all proteins get mutated. So that's my concerns, is like, is it the machinery also doesn't work in
yeah.
We have
real
a
ALS?
Sorry, we have a similar question that among the pathogenic genes, how many of them do we have in animal model? I think it is similar or related. So if you
Yeah, so we have there are some mice models for ALS. Unfortunately, like C9 does, they do have like a working work in progress mice model, I would say, but it does not show all the symptoms of ALS. Like many mice models do not have like motor defects, and they would have like maybe like a C9 defect, a specific defect, but they would not have all these motor defect functions. So that again goes back to, do we actually have like a good model? Um, so
And yeah, no, I'm just another question. I'm just like, as you mentioned that I know that ALS patient slowly they paralyze by themselves, right? Slowly they lose all the motor contact. So do you have any uh, control, like which like any stepwise manner? Because you mentioned that stepwise the motors they lose all motor uh, functions, especially in the. I'm sorry, I'm not able to
yeah, so
maybe.
these are like really rapid progressing disease. So the like within matter of like months, a person might go from having like minimal symptoms to being like completely or like on a wheelchair and unable to walk. So it's that fast. So it really depends on like the gene, like patient and the gene mutation that they have. Some patients are more like slow progressing. They might have these symptoms for like 4 5 years and still be like not as affected, but some patients might really rapidly develop, and within like matter of few months they might even lose their lives. So it really is widespread in that way. So it's difficult to like really pinpoint and say like this is the it occurs like in a more stepwise fashion. This happens first, and after like um, uh, 5 months this happens. I think it's really patient dependent.
So we have a question, Aparna, that you mentioned that it is predicted that there is increase 70% and up to 2040 it will be increased. Is there a specific reason to say that it is increasing?
Yeah, I mean, I just think it's like everything that's happening around us and the way that we are living our lifestyle right now. I really don't know why they predicted that to be 70%, but I'm just like guessing here that it could be just the way that we are having like more processed foods right now, or like not exercising as much, or like having, I mean, it could also just be like genetics over time. Yeah, it just really depends, but I'm not exactly sure why they came up with that number as 70.
Yeah.
Let's hope the number of ALS patients increases because of treatments that will actually work, which will slow down the disease so much that they'll live for a very long time.
Mhm.
Yeah.
Well, thank you, Aparna. It was
Yeah,
quite
thank you.
informative. We learned a lot. And if you have any further question, you can reach out to Aparna directly. So, Oscar. Oscar, you want to introduce the second? Okay. We could not hear you. I think you're mute.
Okay, so thanks, Aparna, for your wonderful talk. And our next speaker is Katharina Meijboom. So Karin is from Netherlands, and she completed her bachelor's and master's degree there. She did her doctoral study at the University of Oxford with a thesis titled Spinal Muscular Atrophy, Disease Mechanism and Therapeutic Approaches. She then joined UMass Medical School and worked with Dr. Christian Muller at the Gene Therapy Center on gene therapies for central nervous system diseases. Later, she became a postdoc in Dr. Bob Brown's lab, focusing on gene therapies of motor neuron diseases. During the COVID pandemic, when Karin was unable to work in the lab, she discovered a passion for hand-on activities like sewing, crocheting, woodworking, and baking. But she is extremely passionate about research and developing gene therapies for life-threatening genetic diseases. So, Karin.
All right, let me start sharing my screen. Thank you for this awesome introduction, and hello everyone. Thank you so much for coming to these talks. And also I would like to thank the outreach committee for inviting me. I'm very excited to share my research. So the bold approach to tackle ALS that I want to discuss is gene therapy. Also, let me quickly ask my husband to lower the volume because I can hear myself. All right, sorry for that. So Aparna has already given an awesome introduction on ALS, so I can keep it very short. But what I want to talk about is gene therapies for ALS. So as Aparna said, ALS is a motor neuron disease that affects movement and in the end also the ability to breathe. And as of this moment, there is no cure or no medication that significantly slows this disease. So the exact cause of ALS is for most patients not fully understood, but research points to a combination of genetic, environmental, and possibly lifestyle factors. So there are currently about 30 genes found that when they have certain mutations, they can cause ALS. And Aparna already showed them in a different graph. So a mutation is a mistake in the genetic code, and I will explain later how that works. So the first gene that was found that can cause ALS when mutated is SOD1, and it was found by my supervisor and his group, Dr. Brown. And in 2016, NEC1 was found, and it was found amongst others by Dr. Landers, which is Aparna's supervisor. So about 10% of ALS patients have a family history of ALS, meaning one of their parents or their grandparents or another close family member also has ALS. So these patients have a mutation in one or more of their genes that causes ALS. The remaining 90% are called sporadic patients, meaning they do not have any family members with ALS. But for most of these patients, we do not know what the cause of ALS is. However, for about 11%, as you can see here in this table, we know that the cause is a genetic mutation. So they did not get these from their parents, but sometimes mistakes in the DNA happens spontaneously. And when it happens early in the development of a person, then it can cause ALS later in life. So let's now talk about mutations. But first we need to know what DNA and genes are. So DNA is like a cookbook. It contains information about every product that you could make. And DNA is like the genetic material. It consists of the genes. And then you have RNA, and RNA is like a recipe. It is how to make a particular product. And then we have proteins, which are the cupcakes or the product itself. So genetic mutations in the DNA are like mistakes on a specific page in the cookbook. So, and if that mistake in the cookbook is on the page of the recipe, then this recipe will not be correct anymore. And when the cell will try to make the product from this recipe, it either won't work and there is no product, or in this example, there will be no cupcake. And we call this a loss of function. Or the protein will be different and therefore toxic and will make the cell sick or die, which in our example would be a terribly tasting cupcake that would make you sick. And this we call gain of function. So here I have an overview of the most common types of mutations. So when we go back to our example of the DNA being like a cookbook, let's take a part of the recipe on how to make cupcakes as an example. So in the original, it says beat eggs for 6 minutes on speed 8. But then if we have a missense mutation, it is like having a typo in a recipe where one ingredient is swapped for another. So then it says beat sugar for 6 minutes. And then for an insertion, we have a piece of information that is randomly inserted into the DNA, or in our example, the recipe. So suddenly you are beating the eggs on speed 908 instead of 8. So then a deletion is just a part of the genetic code is missing, or a part of the recipe is missing. So then we have a duplication, and Aparna already touched upon this when she was discussing the ALS causing gene C9orf72, where there is this repeat that is duplicated many, many times. So in this example, it would be BDX for 66,666 minutes on speed 8. And then there are nonsense mutations, which are like typos in those instructions that tell the cell to stop building the protein before it is finished. So now let's discuss a couple of ways of how to use gene therapy to treat ALS. So when mutated, some of the ALS genes have loss of function pathology, whereas others have gain of function pathology, and some of them have both. So when we go back to the example from 2 slides ago, some mutations result in no or not enough protein, or as in the example, no cupcake. And when we lack this protein, specific nerve cells will die and we will get ALS. So the solution to this problem seems to be incredibly easy. You are missing a healthy copy of a gene, and you're therefore missing the protein or don't have enough protein. So therefore, the solution seems to be then just add a gene or add protein. And well, if you were to just, swallow a bunch of protein, that would not go to our cells and like how we need it, or the cells that we need them to go to. And even if we were to inject them in our blood, it still would not go to our nerve cells. And so we need the DNA to really be changed within the cells. And it's hard to do that. However, one of the most exciting advances in modern medicine has been the discovery of how the adeno-associated virus can be used as an effective delivery system for therapeutic genetic material into living tissues. So adeno-associated viruses, or AAV for short, they are viruses that don't make humans sick, and they also cannot make more of itself like normal viruses can. Like, for example, COVID-19 or the common cold virus. They can replicate many times and spread. And we don't want that with the treatment. We just want a treatment just for one person. So that makes AAV a pretty good option. So we can modify the AAV and take out the virus genes and add genes that we want to deliver to the cells in the body. And this is really cool. And only 7 years ago was the first AAV treatment approved by the FDA. And to date, multiple AAVs have been approved for a range of diseases. And I think the one that I personally am the most excited about is Zolgensma. It's an AAV for the disease spinal muscular atrophy. And I used to work on SMA, not on this treatment, but SMA is a very rough disease, which is also a motor neuron disease like ALS, but it's in children. And where children normally would die after a couple of weeks after birth, now they are living for years already. So there's a lot of promise in AAV gene therapy. So I've been using this technique to find a treatment for ALS. And one of the genes that cause ALS when mutated is NEC1. So ALS patients with NEC1 mutations get sick because of loss of function. And to treat this form of ALS, we have designed AAV viruses that contain a NEC1 gene. We cannot just test this approach in humans because we don't know if it's safe yet. So therefore, we first test this in cells, in cell culture. So we infect this virus into the cells. And when we are working with an AAV, we call this transduce. But it's the same thing, infecting, transducing. So, and after a couple of cells, I took the cells, I harvested them, and I looked if there was an increase of NEC1 gene product. And we did see an increase of NEC1 gene product. So then the next step is to see if this would also work in a living organism. So we have mice that have NEC1 mutations, and these NEC1 mutations cause them to die at a younger age. And also these mice are very small because these two mice that you see here, they were born from the same mother, the same father at the same date. They are brothers. They're both the same gender, but this one has NEC1 mutations, and this one is a healthy mouse. So our plan, and we haven't done this yet, is to treat these cells with the AAV virus and see if that will make them survive for longer, hopefully as long as the wild-type mice, and also if that will help them gain weight and be more healthy in general. But this is still a very much ongoing work. So another strategy is gene correction by editing the DNA. And for this, we can use a technique called CRISPR-Cas9. And CRISPR-Cas9 is a groundbreaking technology that revolutionized scientific research. So I'm wondering if anybody knows who invented CRISPR-Cas9 editing. So Therefore, is it number A, Albert Einstein, B, Anthony Fauci, C, Marie Curie, D, Emmanuel Charpentier and Jennifer Doudna, or E, James Watson and Francis Crick? Well, I see that most people have it right. It is indeed Emmanuel Charpentier and Jennifer Doudna. They are two kick-ass female scientists who won the Nobel Prize a couple of years ago for this invention. So, yes, 95% had it right. All right, so let me go to the next page. Here is a little, I can't start it. Wait, can I start like this? Yes. Here is a little animation on how CRISPR-Cas9 DNA editing works. So for example, if we think of the cookbook example, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it within our DNA. And for this, we use CRISPR-Cas9. So CRISPR is short for clustered, regularly interspaced, short palindromic repeats. And CRISPR consists of two components. There is the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA that we want to edit. So the first thing we want to do is identify the mutation that causes the health problem. And then we can create a specific guide RNA to recognize that particular stretch of DNA. So the guide RNA is attached to the cutting enzyme, Cas9. And when this complex is introduced, and then this complex, we introduce it together to the target cells. It locates the target letter sequence and then cuts the DNA at that point. And then at this point, you can edit it and do various different things. But I'll discuss one particular thing. So I'm using CRISPR-Cas9 to correct genes for a couple of projects, but I would like to highlight one particular project. As Aparna mentioned before, the main genetic cause of both sporadic and familial cases of ALS is mutations in the gene C9orf72. So when we take a look at the gene, we see that 6 letters of the DNA code is repeated multiple times. And that is also the case in healthy individuals, but there it's only repeated up to 23 times. And most of the times it's up to 11 times. But in ALS patients, this can be repeated thousands of times, although a smaller number of repeats can already cause ALS as well. So researchers have found that when this code is repeated many times, it causes both a gain of function and a loss of function. So not enough protein, the loss of function, and a gain of function because this repeat is toxic and causes cells to die. So therefore, we designed different CRISPR guide RNAs to cut out this repeat and thereby correcting the gene. So we designed 2 guide RNAs before the repeat and 2 after, and we tested which ones worked the best. I found through looking at the DNA sequence that combinations 2 and 3 and 2 and 4 work the best in cells. So I continued working with these. And we next tested this treatment in mice that have the human C9orf72 gene with 500 repeats. And we looked at different aspects of rescue of pathology after C9orf72 correction in these mice. But because of the time, I will only discuss RNA foci. So as Aparna mentioned, there is this repeat can cause this RNA pathology, which are RNA foci. So they have these clumps of RNA molecules that I stained, and here you can see them in red. I circled them, and the red dots are the RNA foci, and this is here in brain cells of mice. So when I treated mice with guide RNA 2 and 3 and guide RNA 2 and 4 and Cas9, we saw a strong reduction in the number of RNA foci. Here you see one here and there's one there, but by far not as many RNA foci as is shown in the untreated mouse. We also looked at cells that were taken from an ALS patient. So they were also taken from skin cells, and those skin cells were turned into neurons. And this ALS patient has 450 repeats in the C9orf72 gene. And again, we looked at different aspects, but for now, I would like to focus on only if we were able to rescue loss of function and if we could increase the C9orf72 gene product. So as I explained earlier, DNA is like a cookbook, and I fixed the mutation and got rid of the repeats. So therefore, we wanted to know if the amount of correct RNA, which is like the recipe, was increased. And we saw that that was indeed the case. And according to our example from earlier, ultimately, we want to restore the amount of protein, which in the example is the cupcake that is made from the recipe. And as you can see here, that almost doubled after fixing the DNA with guide RNAs 2 and 4. So this brings me to the end of the presentation, and there are many, many more gene therapy approaches that I was not able to discuss because of the time, but I want the take-home message to be that gene therapies are a very promising strategy for many genetic diseases. But I do want to offer a note of caution, since this is still a relatively new field. Determining whether something is truly safe, especially when using viruses and altering DNA, it requires extensive research and time. However, ALS is a debilitating and deadly disease, so it needs a bold approach to push us closer to a cure or an effective treatment. So I would like to acknowledge my lab, the Brown Lab and the Landers Lab, with whom we collaborate on the NEC1 project. And of course, I would also like to acknowledge all the funding sources. And thank you so much for listening. And are there any questions that I can answer?
Great. Thank you, Karin. That was a very lovely talk. I learned a lot. So if anyone has questions, feel free to type them in the chat box. So I see there's one already. How do the viral vectors pass through the blood-brain barrier? And perhaps you can give a brief introduction to the blood-brain barrier as well.
Yeah, sure. So the brain is a very sensitive organ, and we want to protect it as much as possible, or the body wants to protect it as much as possible. So therefore, there's this barrier around the brain that causes all kinds of stuff to not enter the brain. So that is great for us because we don't want to have any infections in the brain and that sort of thing. However, it's also bad when we are trying to design medications that we want to treat the brain or the spinal cord, because this is all protected and it keeps everything out. However, like I said, viruses, they are so good at infecting cells. And therefore, when you're looking at the different AAVs, there are some AAVs that are really good at infecting the liver, others are better at muscle, for example. And then there is also a couple that are better at penetrating the blood-brain barrier and delivering the treatment into the brain or in the spinal cord, into the motor neurons. But it is definitely one field that lots of people work on because we want to make this as good as possible. And sometimes the other thing that you can do, actually what I did with the mice is actually inject the treatment directly into the brain. But ideally, we would not be having to do that. Ideally, you just give someone an injection inside, an IV injection.
Great, thank you. Our next question is, any thoughts on targeted nanotherapeutics?
Oh yeah, they're awesome. I am definitely very interested in this. The problem, so it's not my field, so I don't know as much as other people know about this. The thing is, however, we're not there yet with nanoparticles. Like there's again a lot of research going on with this, and I think it's very promising, and I'll definitely keep my eye on these. Because AAV, as good as it is, it's also not perfect because it can cause a range of different problems. But yeah, I spoke actually a couple of months ago with someone at UMass who does work with this, a lot of work. She's a professor at the neurosurgery department, and she told me that she thinks it's going to take at least another 10 to 15 years. So.
Thank you. The next question is from Mina. What is the next step when designing gene therapies for ALS? What kind of testing must be done to ensure it improves the mice or humans? And I have a similar question to that, maybe to tag on along with that. Have you seen any improvements in symptoms in mice that you have treated with the AAVs? And you may have mentioned that already, but I missed it.
So to answer your question first, as Aparna mentioned, because like the treatment that I, like finished, or like we have published a study is the one Batsinan or 72 that I just showed. And in these mice, there are a couple of mouse models that have the repeat expansion. So this repeat in like that, and the mouse model that I've used actually 2 mouse models that have the repeat expansion. And both of these mouse models have a molecular phenotype, which means in the cells, they have the gain of function pathology, they have the toxicity caused by the RNA, they have the toxicity caused by the extra proteins, but this mouse model does not have any muscle phenotype. So that means they don't have, they seem perfectly healthy on the outside. So, and they have a normal lifespan, they live a normal life. So it's hard to see if you actually improve these mice. However, I'm currently working on another mouse model, and actually the worst mouse model for this is a mouse model that expresses a mutation in the SRT1 gene. And I have not treated this mouse model yet because I'm not that far in the project. However, many other groups have worked on this mouse model, and they show an incredible increase of lifespan, and mice are better, they can easier move for longer time. It really it takes much longer for these mice to get sick. Oh, sorry, what was the other question?
Yeah, the other question was, what kind of testing must be done to ensure it improves the mice or humans? Basically, what are the next steps in designing these gene therapies for ALS?
Well, so for me, I think then the next step is to personalize gene therapy a little bit. And, because you, on one hand, want to create gene therapy that works for as many patients as possible. But sometimes that is a little bit of a dirtier approach. And sometimes, so therefore, I'm working on more precision gene therapy in which we are fixing mutations instead of getting rid of everything or just adding new protein. I'm not saying that, yeah, I think that is part of the future. Other things that need to improve is, for example, being able to penetrate the blood-brain barrier, just so treatment is going to reach the brain better. So yeah, these are things that people are working on.
Thank you. Another question. For the CRISPR, how do you test the off-target effects of the editing?
Yeah, that is a good point, because when you're using CRISPR-Cas9, you are changing the DNA. And when you change the DNA, that is a permanent thing you do. So you want to make sure that when you're changing the DNA, you only change the part that you mean to change. And you really want to make sure that you're not changing anything else in the DNA. So when we are working with CRISPR-Cas9, we have to look at all targets and see if it's safe. So there are various different tests that you can do. Yeah, so I'm planning to do this and especially in collaboration, because honestly, that's not my expertise, but they are incredibly important.
Yeah, follow-up question about that and safety. So, what are the risks associated with viral vectors?
Oh, a very important risk is an immune response, because it is a virus. So our bodies are, even though this virus doesn't make you sick, your body still wants to attack this virus. So if you have already been in contact with this virus, then your body remembers that. And therefore, when you get treated with this virus, it will immediately try to attack this virus and get rid of it. So that is, again, a reason why we might want to look into other ways of treating and using gene therapy, for example, with nanoparticles. But we are not there yet. So therefore, for now, we just have to work with the AAV, and there are some things you can do to lower this immune response, but it's definitely a risk.
A follow-up question on that. Is it possible to test patients perhaps ahead of time to see if they've been exposed to this particular AAV before and if they would mount an immune response to it? I know that's like very far, or maybe not that far in the future, but to sort of see if this treatment would work for them or if their immune system would just, get rid of the virus, the gene, the therapy virus. So another question is, how are you delivering Cas9 to the brain? Karin, can you hear us? I think we lost Karin. Hello? Karin, hello, can you hear us?
Karin, can you hear? Oh, I think she.
Oh, she said her audio's cutting out. I think she's probably trying to exit the Zoom meeting and come back. Okay, we'll give it in a minute. In the meantime, if everyone could please take a minute and fill out our survey. So I'm gonna post it in the chat box now. That would really help us improve our events. So while Karin is coming back, please take a minute and fill that out. Also, I'm going to reshare our social media links. So if you haven't joined our social media pages yet, please feel free to do that now.
Can everybody see me?
Yeah.
Yes, we can see you and hear you. So sorry about that.
No,
Yeah.
I'm sorry. I have a relatively new computer and I think it's just
That's
I don't
okay.
know.
We have a couple more questions to get through, if that's okay. So, how are you delivering Cas9 to the brain?
Oh yeah, for this we also used AAV. So you
Okay.
can use AAV not only just to bring in a new copy of a gene inside the brain, but you can also use it to deliver the Cas9 gene and also the guide RNAs.
And so are you delivering the virus directly to the brain with an injection, or is the virus traveling to the brain?
No, so for this study, I directly injected it into the brain.
Got it. Okay, thank you. And also, since ALS is caused by mutations in multiple genes, how many genes can be corrected using CRISPR-Cas9? It's a great question.
Yeah, I mean, that is a good question. Sometimes if there's just a really large deletion, I guess you can use prime editing, which is a form of Cas9. But sometimes CRISPR-Cas9 is just not the answer to the mutation. It really depends on the mutation. So sometimes the best way is just to add a new copy to the gene, and then it's just easier to do that through like AAV gene augmentation. And also sometimes you just really want to get rid of as much of a certain gene product because it's toxic. And for that, you can use CRISPR-Cas9. You can just mess up the gene using CRISPR-Cas9, or you can correct it. However, but there are other ways to do it. For example, there's something called antisense oligonucleotides or microRNAs, and these are all different strategies to lower the RNA, so the gene product.
Great, thank you. Last call for questions, and feel free to unmute yourself as well if you have a question.
And also, if anybody has any other questions, that feel free to email me. I should have put my email on here, but my email is Katharina, as spelled there, .meijboom@umassmed.edu.
We can also type it in the chat box.
Oh, that'd be great. Thank you. I'm on the
Yeah,
phone
you're
now.
welcome. You're welcome. All right, thank you so much again. And Oscar, I'll give this
So
back to you now.
Yeah, thanks for both speakers, and we had a wonderful session with wonderful presentations and wonderful question and answers. So thank you again, and thank you for the audience for joining in. And finally, we very much appreciate everyone who joined in today and to help us fill out our survey, which is already in the chat box. And also, if you're interested in sharing this session with your friends or you want to review it, this session will be uploaded to our YouTube channel. So thank you all and see you at our next event. Have a good evening.
Thank you everyone for joining.